Welcome to the SB Grid YouTube channel. Software tutorials by developers, lectures by structural biologists, unique content brought to you by SB Grid.
I hope you're all excited for today's talk from Shores about the new developments in Reliant 5. Just before we get going, I'll mention that our next webinar in a month's time is from Ryan Tripp from the RF Diffusion team at Columbia. So if you're interested in that kind of workflow, a little bit less CryoEM based, then come back for that next month. And just some kind of basic housekeeping, because we are quite a large webinar today. The people who want to see this, rather than doing our usual questions via chat to the hosts, instead we'll be using the Q&A tool this time. So if you have any questions, use the Q&A tool at the bottom of the screen, and at the end, either one of the hosts will pick these up and give them to Shores, or Shores can go through them as well. So since it seems like most people are here now, I would just like to first thank Shores for coming and agreeing to do this webinar, and then without too much further introduction, say that we have Shores Cherez from the MRC LMB, and he's going to talk to us about the developments in Reliant 5. We're really excited about this. I know we're very heavy users of it, and I imagine many of you are as well. So with that, we are now recording this session, and all the questions will be at the end. So take it away, Shores.
Thanks, Sean. Also, thank you very much for the invitation to speak. It's always good to speak in your webinar series. So I thought that for today, I'd just give an overview of new developments in Reliant 5. Before I do that, I would like to show you the current Reliant team. Takanori Nakano was a postdoc in the lab. He left a while ago, but he's still up there because as you'll see, he's still very active on the mailing list and very helpful with debugging, etc. Dari Kumanius has now also left the group, but and he's becoming his own. He's starting his own group at the Charles Zuckerberg Imaging Institute. Johannes Schwab is currently a postdoc in the lab, and Kiraz Jamali is a PhD student. And then recently, Bogdan Todor joined as a postdoc jointly with my lab and Tanmay Bharat's lab. And Zdenka Zivanov, who was a postdoc in my lab, has now left, but has still contributed to some of the tomography stuff I'll show you today. Alistair Bird was a postdoc with David Barfoot here at the LMB, and he's currently working at Genentech with Dmitry Tegunov. And Rangana Varshamanake was at the LMB in the group of Garib Murshidov, but he is now working with the CCP-EM team here in the UK, and where he is responsible for some of the tomography pipelines in the Doppio and the CCP-EM pipeliner. So besides that, we collaborate with all kinds of people around the world, including Eric Lindahl's group in Sweden. So we went beta last year, and as you see, the colors are now blue instead of the previous red or so. And I thought I'd go through some of the new developments, and these are the main ones here. There is all kind of small things around it, but these are the main new features in Reliant 5. So there is a new regularization approach for any type of refinement or classification that was done by Dari. Johannes has written Dynamite for treatment of continuous heterogeneity. Kiraz has written Model Angelo for automated atomic modeling in cryo maps, and also for identification of new proteins in maps. And then Alistair has written some Python software around Reliant 5 that enables us to do a whole tomography pipeline, and then Bogdan and Rangana have been working with this pipeline and further developing it. So I will not speak today about ModelAngelo because I understand Kiresh already spoke in your webinar series, and I guess that was recorded too. So do have a look there. I'll only mention that you can now run ModelAngelo from Relan 5. It's just a atomic modeling job type. And just to say that if it gives bad results, make sure that you're not trying to build in the inverse of the map. So you may have the wrong hand, which could of course happen in cryo-EM reconstruction, or make sure that you provide the correct FASTA file with the protein sequence that you're building. If you do those two things right, then anything up from 3.5 angstrom resolution should give you a pretty good model to then start your own modeling in Qt and refinement. Good. Let's start then with the regularization, a new approach for regularization by Dari, and he called this a blush regularization. So I think one of the main successes of Relan has been the explicit formulation of the cryo-EM reconstruction problem in this Bayesian framework, where we combine likelihood function that describes your forward model with an explicit prior in the and that expresses the prior knowledge you have about the model in the absence of any data. Now, in the absence of any data, it's of course quite hard to know what your cryo-EM reconstruction would look like, and standard Relan has therefore used a relatively information poor prior, just assuming that the densities in real space are smooth, or it's actually expressed in Fourier space, where the power of Fourier components in the reconstruction are kept low in the absence of data. Now, the advantage of that is that you get if you express the entire problem in Fourier space with this type of prior information, you can then analytically solve for the regularized likelihood function and optimize it directly. And that algorithm has been very successful, of course. But of course, we do know a lot more about the reconstructions that we're going to make with Relan. Even if we haven't solved the structure yet, we probably know it's made up of proteins or nucleic acids, and the amino acids in the protein form chains which fold up in secondary, tertiary, and quaternary structures, all of which we have information about from the PDB from many decades of structural biology attempts. But of course, how to express this information in a mathematical function that you can then optimize is far from easy. And that's where Dari's development of Blush comes in. So that rather than having a explicit parameterized model like the one in Fourier space in the old version, Dari decided to try and use a neural network, specifically a denoising neural network, to express the type of information that we get in previously solved cryo-EM reconstructions. And then, of course, if you know you have a protein, you could have a neural network that does some sort of operation on your cryo-EM map that makes it look more like a protein. And there are softwares out there that do exactly that. And some people have even proposed to use these type of networks inside the iterative reconstruction algorithm, for example, of Relan. Now, there is a danger in that if you do this too aggressively, then you might start to hallucinate protein-like features, and you then kind of inject these into the refinement, into the iterative refinement algorithm. Then this may become ever worse, and with the outcome that the reconstructions you get out might have invented protein structures that were not really present in your data, but you've somehow injected them too aggressively in your reconstruction algorithm. So to circumvent that problem, Dari set out to use just unfiltered half maps that he downloaded in pairs from the EMDB, and sifting out kind of redundant structures, etc., or take away too many ribosomes, etc., then he was able to make a set of about 400 half-set pairs. And then he trained a denoiser using a noise-to-noise, basically a noise-to-noise framework. He made several adaptations of that framework to how to deal with, for example, with explicitly deal with fuzzy regions of membrane patches, etc. But you could think of it as a noise-to-noise type of denoiser. And then this denoiser is put inside the iterative algorithm of ReLAN, where every iteration we have two independently refined half maps, which are denoised by this denoiser and then used for the references in the next iteration. And it replaces the kind of Fourier Wiener filter that was the result of the Fourier-based prior in standard ReLAN regularization. And in the figure here on the right, you can see just if you give it one input map, a single execution of the denoiser here in panel D goes from this type of input map to this type of output map. So you'll see that any noise type of things in the solvent get flattened, this membrane patch gets flattened nicely, and then density inside the protein region is mostly retained, but perhaps sharpened up a little bit. And this panel E here is another example. Now, if you do this iteratively, so even though at each initial step, the changes are perhaps not that drastic in the density, they're very subtle changes, but by doing this iteratively during the, say, 25 different iterations that a typical refinement might take, you get quite drastic results, different results at the end. And one of the examples we showed this for was this PFCRT data set, which is you can download from Empire. It's a 10330. And if you look in panel A, then colored by local resolution is the result of a standard ReLAN refinement if you process this data set. And because of variations in local resolution, etc., you get overfitting of regions in the solvent and the detergent and parts that are floppy. And because the blush regularization, this denoising network is quite good at smoothing these out, then the final resolution with blush is of much higher resolution. And we now measure this not only by FSC curves, which you can see here on the right, but also by how well our automated model building program, ModelAngelo from Keras, is able to build in the blush map compared to the original map. Another example is this GPCR dimer, C2, which has relatively little density sticking out of the detergent micelle. And again, standard ReLAN then gives a rather suboptimal map where alpha helices are basically just tubes, whereas blush regularization makes the map a lot better, both in resolution, but also what ModelAngelo can then build in it. Now, this is not only something you can do in 3D auto-refine, you can also use blush regularization in 3D classification, or in this case here on this slide, in multi-body refinement. And this is the typical spliceosome data set that we use for multi-body refinement. We use this for multi-body refinement introduction. And the map here in panel A is that multi-body refinement using standard ReLAN, colored by local resolution. And then panel B is with blush regularization. So everything is the same, but we apply blush regularization on each of the bodies at every iteration. And that again leads to drastic improvements in FSC curves for many of the regions, and also the completeness of the models that you are built by ModelAngelo. You know, the SF3B domain would still be relatively poor density in standard ReLAN. Here, local resolutions are around 7. So ModelAngelo would build hardly anything in it. But using blush, it then goes up to here this type of resolution, close, not quite, but close to 4 angstroms, and then Model-Angelo can start to build a considerable bit of the structure. Now, another example is from our work on amyloid filaments. So this is quite a small filament. It's made up of only a dozen or so residues in 2 opposing beta strands, which pack against each other. And that makes that the images are relatively no signal to noise, and standard ReLAN has big problems in doing this reconstruction. And in this particular refinement, we couldn't even separate the 4.7 angstrom distance between the individual beta strands in the beta sheets along the helical direction. But the very same refinement with blush gave a beautiful model, which Model-Angelo built completely in it. Then the last example, this is a 40 kilodalton protein nucleic acid complex. That's how it was called in the original preprint. Now we know it's this ACA2 complex. So it has a bit of nucleotides here, which of course make signal to noise ratio a bit higher than if it were just protein alone. But using either ReLAN 5 or BryoSpark, and this work was done by Max Wilkinson, Max was unable to solve this structure at all, and he then sent the data to Dari to try with blush. And Dari first took an initial model, did some 3D classification and a refinement, made a new initial model because the one he first made was kind of preferred orientation, then did another 3D classification, put things through polishing and then refinement. And then from 300,000 particles or so, he got this beautiful map to about, I think the overall resolution is around 2 point something angstroms or so. And then Model-Angelo can build the entire structure relatively straightforward. And each of these purple bits of the processing pipeline, Dari used the blush regularization. So I'd say in conclusion of this part, blush regularization can make your structures a lot better, especially if you have low signal to noise ratios and then your refinements are suffering from various types of overfitting. So kind of seeing kind of sunshine ray-like features coming out of your structure because of low signal to noise ratio. Those are the kind of things that blush is very good at removing and therefore making much better refinements. So that's what happens for these small proteins like the ACA2, or for example, for the multibody where the individual bodies get relatively small too. Cool. Then the next topic is dynamite, and dynamite is written, as I said, by Johannes Schwab, and he came up with this name, which stands for Modeling Structural Dynamics that might improve your map. And I think with the latest version, it might also be called Dyname, because I think the might is better to, we're now more confident in its capabilities. So this is a variational autoencoder type of network, which is trained, we trained 2 independent variational autoencoders on the 2 half sets of our data set. So I think that's a first in this type of VAE approaches, where we keep everything completely gold standard in terms of separation of the 2 halves. And then the type of VAE is we've leaned heavily here on the E2GMM approach, which was originally introduced by Muyuan Jen and Steve Lutke, which we also use a variational autoencoder to move around a 3D Gaussian model of the reconstruction. So in that sense, the dynamite is very similar. We have an encoder that maps individual images into a latent space, and then a decoder which predicts deformations on these 3D Gaussians. So these deformations are then applied to a consensus model of the Gaussians, and then using a forward model that includes projection operator and the CDF, it's predicting the image, which is then compared to the original image, and that loss function is optimized when you train the VAE of the whole data set. Now, besides that, and that's kind of then also where the similarities with E2GMM stop. Besides that, Johannes has also implemented a weighted back projection approach where he estimates a inverse of the displacement, so a 3D deformation field which kind of does the kind of moves Gaussians back from the positions in the individual images to the consensus, and he then uses this inverse deformation field to do a weighted back projection of the original experimental images, where now densities are displaced in 3D according to these inverse deformations, in an attempt to make a consensus reconstruction, which is kind of made from morphed images where the densities that were moved away in the images are kind of tried to be placed back. And because we do this completely gold standard, we have now two independent unfiltered consensus reconstructions, which we can compare with each other using standard FSCs and post-processing procedures. Now, one thing I didn't say is that the main reason why we chose to work with displacements of 3D Gaussians in real space is that you can now impose regularization on the deformation fields, because there are plenty of parameters in the variational autoencoder, so many that we were worried that the problem again would be very ill-posed, just like you have the refinement problem in cryo-EM is ill-posed, where many different orientations can give rise to the same, where many different reconstructions can explain the data equally well. We were worried this would happen here too. And therefore, we would need to, again, inject prior knowledge to select the correct solution of all those. And we thought that doing that in real space on these 3D Gaussian deformations would be a straightforward thing, or kind of the most logical thing to do. So again, this is the same spliceosome as I've shown you before for the blush data set. This is just to show what the different types of regularizations do. So in panel A is standard consensus refinement of ReLiON without multibody or blush or whatever, just a standard refinement of ReLiON and colored by local resolution. And you can see that the moving parts, some at the bottom here, but especially here at the top, have relatively low local resolutions. Now, panel B is the deformed back projection map combined from two halves of dynamite without imposing any regularization on the Gaussian. So every Gaussian can go in whatever way it likes. If you don't use any prior knowledge, then you can see that you even make things worse than they were in the consensus refinement. So dynamite without any regularization on the Gaussians doesn't work at all. If you now use the because we've solved these structures using multi-body and people have built models in them. If you take these atomic models, rigid body fit them in the consensus map, and then use a coarse graining of Gaussians on top of these atomic models, where you keep distances very similar to what they were in the atomic model, basically making the atomic models move around as partially rigid, as partially rigid bodies, then you get, and you run that in dynamite, you get this reconstruction. And then panel C is an intermediate between no regularization at all and this very strict regularization based on atomic models, where we just fill in panel C, we've just filled the map with Gaussians kind of randomly just to fill the whole space. And then we apply elastic network type of regularizations, which keep Gaussians together, but also keeps them from bumping into each other or some Gaussians becoming too lonely. But there is much less prior knowledge in terms of all the atomic models that went in panel D. But you can see that you can still get a very good reconstruction. The reason we did panel C and not just panel D and called it a day was, again, that we were worried about model bias injected from atomic models into the regularization. And we actually tested that for, if you take the SF3A domain, which is very flexible, you can put the correct model and then do a consensus reconstruction in Dynamite, then this is the density of the consensus reconstruction. Oh, I get quite good density back for the model, so that's a good thing. However, in a control experiment, if you use the incorrect model here, then you get density for the incorrect model back. So this indeed shows that there is room in the many parameters of the VAE for extensive amounts of model bias. So any approaches that use explicit atomic models to model these type of heterogeneity in cryo-EM, one should consider that there could be an extensive amount of bias in large number of parameters of neural networks to do this. Now, the more motion there is in these domains, the bigger the effect. So this is the SF3B domain, which is much better ordered. And again, using the atomic model gives you this map, and an incorrect model gives you this map. And the same still happens, but the extent of model bias does decrease. So it will depend also on how flexible things are and how much the signal to noise ratio is in the amount of model bias. But based on these observations, we have decided not to expose this type of functionality in ReLAN. We only expose this type of functionality. So you just work with the consensus map, fill it with Gaussians, and keep them more or less together in terms of priors. Using that approach, we then tested on two different data sets. The first one is a subcomplex of the yeast kinetic core called the CCAN complex bound to the SEMP-A nucleosome. And this is a consensus refinement from ReLAN, low pass filtered according to local and colored according to local resolution. And in D is now the latest version of Dynamite. And those of you who may have seen the preprint of Dynamite a few months ago, this is now considerably better than the preprint. So if you want to use the latest version of ReLAN in the branch, then you'll get the even better results than what we had before. So you can see again a drastic improvement in local resolution, where here the alpha helices of the proteins in the nucleosome were kind of more or less just cylinders, and here you get beautiful side chains popping off them. And that is because this complex is very flexible, and you can just traverse latent space, just like people are used to do with CryoDragon or E2GMM, etc., and make these kind of movies of the type of motions that are present in the sample. And by kind of undoing all these motions in this deformed weighted back projection, you can get these largely improved consensus reconstructions. Okay. Now, the second data set we tested this on was on the full yeast kinetic core complex, again bound to nucleosome. And again, here panel B is the consensus reconstruction from ReLAN colored by local resolution, and panel C now is the result from the latest version of Dynamite. So again, you can see improvement in local resolution throughout, and the more it moves, the better the more improvements you get. However, if you divide this complex up in 4 independently refined bodies, and you do standard multi-body refinement in ReLAN, then you can get a map which is again colored to the same local resolution scheme, looks like this. So of course here you've had to define 4 masks, etc., but you can see that the local resolution in the multi-body refinement reconstruction is still a lot better than in the Dynamite. So I think that means there is potential to make methods like Dynamite still a lot better. Then we sent this to Nature Methods, and then the review come back, and normally we don't like to compare our methods to other people's methods, because many of these comparisons I've seen in the literature don't add much to papers. But the reviewers asked us to compare to existing methods in the field, and 3D Flex on CryoSpark was one of them. So we then ran 3D Flex with default parameters and the same amount of latent space dimensions as Dynamite. And the output of the 3D Flex map of the method, again colored according to local resolution from the two half maps from the 3D Flex method, is on the right, and Dynamite, the map that I just showed you, is on the left. Now, if you just look at the colors, it appears that the 3D Flex map is better local resolution than the Dynamite map. However, if you now look in detail, you'll see that many of these colors don't make any sense at all. So it says this is 4 angstrom resolution here, but there is no information at all. And you know, this is bluer than this, but you can see the grooves, the major and the minor grooves on the DNA here much better than in the 3D Flex reconstruction. So we think we may not be experts in running 3D Flex. This is the first time we've ever run CryoSpark in my lab, but with just using default parameters in Dynamite, the map you get is better, even though it thinks the resolution is lower. And this may be because in 3D Flex, there is a single neural network that does the deformations for the two half sets, and you only calculate the two half reconstructions at the very end from the same deformation model. So it could be, again, that overfitting because of non-standard treatment of the two halves then leads to overestimation of resolution in 3D Flex. Now, just to kind of, as a control to make sure that we're not just very bad at running CryoSpark, we also ran Dynamite on Empire 10073, which is I think this is the TriSNP spliceosome data set. And this is the data set, and this is a figure from the 3D Flex paper on the right here, colored by local resolution as estimated by CryoSpark. Now, we've run this on the same data set. We've run Dynamite. It's a bit complicated because we don't know exactly which particles we're using in the 3D Flex paper, but we've tried to do our best, select a homogeneous subset, and this is the result from Dynamite. So again, the local resolution, in this case, both are estimated by CryoSpark, are better in the Dynamite map. So I think the current version of Dynamite is probably outperforms 3D Flex. Now then, the last new feature in the Rely-On 5 I'd like to tell you about today is now a more or less complete pipeline for subtomogram averaging. And you can, this is now very recent. So when we went beta last year, I told people don't use this yet, and that warning is now over. I think now you can use it, and we'll soon write a preprint and then send a message to the mailing list to encourage people to do it. But you can already more or less safely download the latest version from GitHub now. And then if you do Rely-On -- tomo, you get this GUI, which starts from a tomogram import at the top, which just starts from raw movies from your direct detector and MDoc files from your Serial EM or your Tomo5 or whatever data acquisition software you're using. It then goes through very familiar jobs like motion correction, CTF estimation, etc., which work very similar to single particle analysis, with a few new options for making things better with tilted images. And then Alistair has written wrappers in Python to around RTomo and IMOD for alignment of tilt series. And then we use an algorithm from Jasenko to do reconstructions in Rely-On. And then there is again a program written by Alistair in Napari to actually pick subtomogram or individual particles in the tomograms. And from there on, you can then go to extract particles, do your initial model generation, your 3D refinements, and then use iterative approaches of the tomogram versions of CTF refinement and Bayesian polishing to make reconstructions ever higher. So just a screenshot of the Napari picker. This is actually coordinates that are selected from this HIV immature capsid data set from John Briggs, which we use 5 tomograms of for the Rely-On 5 tomography tutorial. And you can pick you don't have to pick all the individual hexamers on the spheres. You can, Alistair has written an option in this picker to define spheres, and then we just randomly sample particles from the spheres and then sort things out later in classification and refinement. And doing just exactly that, Rangana and now lately also to a large extent Bogdan Toder have used this very approach, and Bogdan has now shown that you can get all the way back to 3.3 angstroms from this small data set of 5 tomograms. And just for comparison, using very carefully picked and selected pre-processed particles that were done by Zunlong Key in John Briggs' lab, he ultimately got 3.2 angstroms out of this data set. So we're still missing 0.1, but kind of giving the much lower amount of effort that went into the particle picking, basically defining a few spheres in 1 hour or so. I think that loss of 0.1 angstrom is acceptable, and the density we get out in the end is beautiful. Michelangelo can build the model straight away. Now, because this is now very new, so we have now just updated the release 5 version of Read the Docs of Relan. So you can now go there and you can download the 5 tomogram data sets from Empire, and then you can go through Bogdan and Rangana's notes and do this yourself for the HIV tomogram data set, and hopefully that will set you up to do this for your own data as well. We will soon add pre-calculated results there too, to help you kind of see where you may have gone wrong or not. And then hopefully we'll write a preprint. Well, we are in the process of writing a preprint, and hopefully we'll post that very soon on bioRxiv, so you can then see how that all works. Just in terms of kind of future outlook for this, as I said, Rangana is now working with the CCPM team to implement all of these tomography procedures in the tomography pipeline of the CCPM pipeliner, where it will then also be accessible through the Doppio interface. So hopefully this, together with Doppio and CCPM pipeliner entries to all kind of third-party softwares, like for picking TomoTwin or whatever, denoising CryoCare, etc., we can then have a marketplace where all different types of software come together in one big tomography pipeline managed and installed by CCPM. Cool. So that brings me to my conclusions. Blush will make your difficult refinements a lot better. Dynamite has become a lot better than its first version, and is now outperforming state-of-the-art in methods in the field that deal with continuous flexibility. And our new Tomo pipeline provides a start to end. You can start from raw movies and end with atomic models built in Michelangelo, which will hopefully make the whole endeavor of subtomogram averaging a lot more accessible for many people who want to try this out. So that's it. Thanks to all these very bright and creative people who I've had the pleasure working with over the past years. Thanks to everyone who sends us bug reports, and especially if they're carefully curated with little test sets, etc., which makes it so much easier for us to hunt for these problems. Just as a final note, all our software development is completely open source, unencumbered by any patents, and we proudly believe that that accelerates science. So I thank you very much for your attention, and I'll be happy to take any questions.
Oh, that was fantastic. Thank you, Sjurs. That was a great talk. So we've got a couple of questions already in the Q&A. I'll just read them out for you for now, and then...
Oh yeah, I can see them now. Is Blush modifying final output map directly in any way, or is it only denoising during iterations to improve the final alignments? So in 3D auto refinement, it only denoises the intermediate iterations and leaves the final half unfiltered half maps and the final combined map intact as it was. During 3D classification, you just run for a given number of iterations, and we never know which one is the last one. Also, the last one will be affected. And then there's a question from Rainandani Kashyap. Could you please explain the meaning of the PCA plot on the left with respect to the changes you see in the Crayam density? I assume that will refer to the dynamite, and the PCA is a low dimensional projection of that 5 or 10 dimensional latent space of that that sits at the heart of the variational autoencoder, and it is a representation of the conformational heterogeneity in the data set, and you can traverse this very much like you do in CryoDragon. Cool. So then there's a question by DA. Were multiple meshes used for the 3D Flex runs, or is it a single unsegmented mesh? We did not use any type of user interaction to design meshes or whatsoever. So we just used the default. Could you please discuss a little bit about the resolution range that you recommend the use of Blush and Dynamite, of the range that they help in the refinement? Yeah, so Blush, I mean, Blush will mainly improve if overfitting is a problem. So it's not so much a given resolution range. If you see the type of artifacts that are standard, that are typical of overfitting, like the sunshine rays that I mentioned, that sometimes you can see that the solvent region isn't flat, but there's actually white and black really extending densities. That Blush is very good at removing, and therefore making those refinements a lot better. So those are the type of refinements where the signal to noise ratio is pretty low. Now, for Dynamite, I think if there is completely no density in the consensus reconstruction, then Dynamite will probably have a very tough time making improvements too. So provided there's already some structure in the flexible paths, in terms of structure, in terms of, perhaps some tubular densities for alpha helices, etc., then Dynamite will probably have a better chance at improving your map than if the domain you're looking for is so floppy that there is hardly any density there. So again, yeah, that's. Then there's Nicolas Vangels. Given that multi-body refinement can still outperform Dynamite, do you see any possibility of combining the two in some way? Yes, and we're thinking about this right now, and hopefully the next version of Dynamite will be better because of it. Then anonymous participant, could you please explain how Dynamite is better over multi-body refinement again? Yeah, so Dynamite uses a gradient descent-like algorithm to optimize the parameters of the VAE, which then lead to the deformations of the Gaussians, whereas multibody does a actual search of orientations of individual bodies. So the underlying assumption, I guess there's two aspects to this answer. One is that the model is now much more complicated, right? So Dynamite has a 3D deformation field for a whole set of Gaussians, and whereas in multi-body, there's only these 4 bodies, each of which have 3 rotations and 3 translations. So the model of Dynamite is much more complex. So you could make the same model in Dynamite, which only has a few rotations and translations of sets of Gaussians, which all move in the same way. And that's kind of what we're currently trying to do. Then the second aspect is that also the optimization algorithm is different. So that's where the gradient descent is capable of getting to the global min, or the better minimum that was found by multi-body angular searches, also remains to be seen. Can you recommend ways to denoise at the 2D class stage? We haven't done that. It's not possible in Erlang. Does Blush help orientational preference problem? Yes, it can. It has been trained with maps that were suffering from preferential orientation type of artifacts. And so it has been trained to recognize those, and it may make those refinements better. Then from Sasha, would you expect Blush regularization to work on graphene data sets? Denoising doesn't always work on graphene. I don't see any reason why not. And, you know, often if there is graphene or graphene oxide data, the signal to noise ratio decreases. So Blush may actually be quite good for that. And then Rashanandani asks, can we use the local resolution map to make a conclusion that the low resolution regions are more dynamic part of the protein and vice versa? Yes, I think so. I think if local resolution is lower, that, well, it could also be absence of, you know, if it's compositional heterogeneity, then that's something different, something that Dynamite will not model, which could also lead to lower resolution. But in general, yes. And also I would say you could interpret the observation if the improved reconstruction from the deformed back projection has better local resolution than the consensus map. I think one conclusion is that at least the 3D deformations that you've estimated in part have to be correct. Otherwise, you wouldn't get that increase in resolution. So to what extent they're correct or not is still a question, but it provides some form of validation of these motions, which of course is very difficult in other programs that just look at the motions themselves. So then Jan asks, have you seen FSC from Dynamite does not drop to 0 at higher spatial frequencies? What could be the cause of that? Yeah, I would say duplicated particles could be, but I don't think we've seen much of it. But if you have a clear case, do reach out. Then Michael asks, does the new tomography pipeline still play well with batch scripting as opposed to interactive GUI? Yes, all the schema, any type of Python scripting you might be able to do with the CCPM pipeliner would all work in the tomography pipeline as well. It's very similar to the single particle pipeline, and hopefully therefore will actually contribute to kind of making things easier where people script other software into the similar pipelines too.
Great, thanks, Charles. I think that's through all the questions in the Q&A, although I can see that Pete has his hand up to ask a question, so I'll let him just unmute himself to ask.
Yep. Yeah, co-hosts cannot dump into the Q&A. So I guess, first off, excellent talk. And second, this is kind of a more general question that you may have perspective on. With the increasing number of neural network-based tools and being used by researchers that don't often have lots of expertise or intuitions about them, do you have any guidelines or rules of thumb for how people should be using these tools to try to avoid causing problems?
Well, I think, read papers and try and understand what you're doing always helps, whatever method you use. But I think that's kind of part of the reason why we already wrote the Dynamite paper. There's still some unresolved problems, I think, but I think they're not just unresolved problems in Dynamite. They're unresolved problems in all of these approaches. And just pointing them out and making people aware of these problems, and ultimately the burden lies on us, the developers, again, to make methods that are more robust and suffer less from some of these problems. And, validation of motions, validation of deformed reconstructions. What is a deformed reconstruction? It is no longer, it's a bit like the combination of individual reconstructions from multi-body refinement, which we've called Frankenstein maps. The deformed back projection map is a type of Frankenstein map that it doesn't really exist. How does the density look like at parts where domains come together, etc., isn't all that clear. So all these questions remain open and will provide food for thought for developers. Hopefully, will be solved in the future, such that the burden will reduce on the user. Is what I would prefer.
Yeah, I was particularly impressed by the section where you had like intentionally put the model in the wrong spot or the wrong model there and do it as a control experiment. And yeah, I don't think many end users actually do that when they're testing out new methods. But
yeah, so in general, I would urge a lot of caution in using atomic models in any type of methods to estimate motion. And a lot of people have started trying to do this, but there is a lot of scope for bias, so be careful.
I am in favor of, in general, doing controls, even if you're doing stuff on a computer. Thank you.
I guess I did have a question just to kind of follow on for that. Do you have any worries about, say, taking maps out of blush regularization and then putting them into these other machine learning neural net type denoising algorithms like your Deep Enhancer, your EM-Ready, things like that, and moving further and further away from the initial data itself?
Whether I have worries about that?
Yes.
I do, yes. And you know, that's what we set out. Blush has been designed to be conservative in that aspect. We do not want to hallucinate proteins. Again, there's a whole potential of getting ourselves into a lot of trouble again by hallucinating proteins. And then especially if you put it inside the refinement loop, then your hallucinations will be picked up as model bias and just enforced and will go and will make reconstructions, which ultimately may again become some sort of new form of Einstein from noise. So we have to be careful there, I think.
You think of any ways
also,
to...
you know, the use of Deep Enhancer or EM-Ready, etc., I think these methods can be useful, but you should just use them at the end to assist your model building. And I would still encourage you to refine and assess the quality of your final build model against the reconstruction from the EM data. But you know, there's of course a little bit of a gray zone there that again will need exploration and will need to come up as developers with robust methods.
All right. Thank you, Charles, for the really excellent talk and the interesting, if somewhat worrying, thoughts about how we're going to deal with these problems in the future. I will.
The future is bright.
I will quickly mention you will not have to pull these Reliant builds from GitHub. We'll be pushing out a new build shortly with all of these new features and developments in Dynamite and in the tomography pipeline in our Reliant 5 beta build in SPGrid. So that'll come out from us in the next couple of days. But yeah, that was really interesting. Thank you again, Charles, and thank you for everyone who's attending. For those who are interested, our next webinar will be in a month.